Great, great. So this is very exciting. Talking with Dr. Wengzhong Xiao. Is that how you say? Is
Yeah,
that
that's
correct
perfect.
pronunciation? Okay, okay. Um, and boy, does he wear a lot of hats. Uh, he's the director of the Immunometabolic Computational Center at Massachusetts General Hospital at Harvard Medical School. Note the computational part. And he leads the Computational Genomics Group at Stanford Genome Technology Center. His research is at the interface of computation, genomics, and medicine. And and I wonder if you could just talk briefly about that, because there's so much data coming out now. And we're going to talk about the severely ill patient study. That study included over 1,000 tests per patient, producing, at least at that time, and perhaps still, the biggest data set ever generated for a cohort of ME/CFS patients. And it included everything from the genome to gene expression to cytokines to metabolomics to the microbiome to cell-free DNA sequencing. So it's an enormous amount of data that is now available to researchers, and you are trying to put it together and make sense of that. Can you? I guess it's an enormous field in itself, just computational biology. Could you talk about that a little bit?
Sure, sure. And it's really great to have this opportunity to speak with you. My background is, as you mentioned, the computational genomics. I started with Ron Davis back, I think, since 2000. The Genome Technology Center that Ron Davis directs, as you know, at Stanford, basic focuses on developing new tools to look at the human genome and apply those tools to study different diseases. So I have had the opportunity to work with Ron for the past, I guess, 20-some years now. And we developed some of those tools together, both on the so-called wet lab side as well as on the computational side. So that's sort of my background, and we together applied, for example, some of these techniques to study patients in intensive care units. As we know, even during COVID, a lot of patients die, unfortunately, after infection, but not directly from the virus, we think, itself, but from our inflammatory response to the virus. So we've been studying that for, I guess, more than 15 years now. And we try to essentially figure out ways to treat patients in intensive care units, sepsis and other similar type of problems. So when Ron started to study ME/CFS, I actually had the chance to know his son before he was ill, as a young, talented photographer. And it just seems to be a puzzle that, as a computational person, I thought we might be able to help in figuring out potential reasons and potentially ways to help him. So I then spoke with Ron Tompkins, who you might also know, at Mass General Hospital, who's a great surgeon and a physician scientist. And he became interested in this as well. So we teamed up with Ron Davis and Open Medicine Foundation to start studying this disease. And as you mentioned, one of the early attempts that we thought we would, start to sort of get firsthand experience in looking at patients would be to select a group of severely ill patients. The idea would be, if somebody has typical, presentation of a disease and it is severe, perhaps the signal are more homogeneous. So that's sort of the experience that we had when we look at intensive care unit patients. So some patients would, got gunshots, some, would hit by a car, some would get severely burned. But when we look at, and some of them would have, infections. But when we look at their, genome or genomic response, they seem to be actually quite similar. So we thought that, in that case, you might be able to find a common drug eventually that can treat a lot of patients. So that's sort of our motivation early on. I think this was like 2016 when Ron and OMF started the severely ill patient project. So, fast forward almost like 8 years, I think we're still using the results that we found from that study. If you don't mind, I can actually show you a slide of I probably need your permission to
Sure.
enable the sharing.
Oh, no, this is new for me.
Yeah, so there's a share, and you would have
There it
to,
is. Just
yeah,
just hit the share button.
yeah,
Share screen.
yeah, to allow sharing.
Okay.
Or I can just send you later. But basically, what we learned is, there's really a number of molecular features that are significantly associated with patient symptoms. Yeah, let me. It still says sharing is not.
Okay, hold on a second.
Yeah, well,
I think
yeah,
I
we can figure this out.
yeah. Are we back? We're back. Okay,
Yeah,
yes.
yeah, yeah.
I wonder,
No, it's okay.
let me see if I can just share photos. Maybe that's what I need to do. Uh,
So it says the
no.
host is not isn't allowing sharing for this meeting. Yeah, it doesn't matter.
Okay.
I can send you
Okay.
the slide,
Some
but basically we
something to
identified
learn.
yeah, we identified a bunch of molecular features, and one of the things that we thought, especially as a computational person, we should do is to be able to validate, those. The reason was, I mean, you know this extremely well that, in the old days, perhaps still now, MCFS is severely, underfunded. So, we were only able to do 20 patients at that time.
Yeah.
So, the significant associations that we identified, we really don't know whether those relationships are robust or not. And but despite of this, we formulated a few hypotheses, and even after 8 years, I think some of those hypotheses are still remain to be tested.
Hmm.
I'm happy to report that some of those later on have been, you know, uh, repeated by other groups. And one example I'm sure is we found that in the severely ill patients, the cortisol level in the morning, to patients, I'm sure it's not surprising, but to us, especially to me, who doesn't really know patients, didn't really know patients at that time, it was a significant signal. And later on, during COVID, there are a number of groups that looked at post-COVID syndrome. Akiko's group and Mark Davis' group, for example, they also showed the suppression of cortisol level in long COVID patients. So that type of validation is what we've been looking for. One of the
Did
another
did did
Mhm.
did cortisol really pop out? I mean, was that like,
Yeah,
wow, it's
yeah,
really
yeah,
low in these
yeah.
patients? Yeah.
Yeah, I'm sorry. If we have the sharing function, I'll show you
Oh,
the slides. But basically, it is the significant one. And there's a paper, I think Akiko's group published last year on Nature. They say that even just use cortisol itself, they were able to distinguish patients from controls at something like a close to 100% sensitivity and
yes.
specificity. So apparently it is, the question is what one, I guess, what that imply. And as a computational person, I want to sort of formulate the hypothesis and test that computationally. And then the second would be, what we can do about it, right? So is it actionable? So those are sort of two areas that we're currently, working on. You probably have also, heard some of the presentations by Akiko. They also looked at, for example, the upstream of cortisol, the HPA axis. But my understanding is that that appeared to be normal. But
Hmm.
then, is the adrenal glands itself, um, you know, having issues? So that type of hypothesis generation and hypothesis testing are things that we're working on at the moment. Another example would be, there's a preprint from Akiko's lab that they showed some of the hormone differences between patients, long COVID patients versus controls in females and males, and that might also support this idea that maybe adrenal gland should be something that we should, look into further. So that's just the example of one of these hypotheses that apparently are supported by multiple studies in MCFS and severe MCFS and long COVID.
Yeah. I saw that in a conference. I was, 2019. I think it was you talked about you found cytokine differences and metabolomic differences and genetic findings, including lower levels of a neuroprotective metabolite in the
Yeah.
tryptophan pathway, which is always which has really gotten a lot of interest over time. Are those things
Yeah.
panning out,
So
or
exactly.
yeah,
So
yeah.
so we're also testing some of those. So you mentioned the cytokines. One of the cytokines that we identified early on is this so-called brain-derived neurotrophic factor, BDNF. And we analyzed, for example, the NIH study that published recently, and we saw the same observation in the muscle of uh, those patients that NIH studied as well. So the short answer is that we're looking for sort of reproducibility among different studies and see whether uh, that is indeed something that we can follow up. So for BDNF, we actually think that there might be something there, and we're still looking for more evidence. The part of the issue with BDNF is when it is in the central nervous system, it's well studied that it is involved in a lot of neurological diseases. Where in a disease state you would have low level of this cytokine, but then in the peripheral system, apparently it has a sort of different goal, especially in females. It would basically control the utilization of fatty acids. So if that molecule doesn't exist, apparently our body wouldn't be able to, in females, switch from using glucose to fatty acids. So that's something that we're currently looking at. So these are terrific questions. I wouldn't say that all of the observations that we identified in the severely ill patients all turned out to be true, but
Right.
that's a reason that we actually need this reproducibility study to know whether those are real leads or not, just because of sample size issue.
Yeah, it sounds like you're being really careful. You're taking the results, you're taking your data, and then you're comparing it to other data to see if it matches up with other studies. I noticed you did that with your latest paper. So you got this huge data. You got all these different, from metabolomics to genomics, and how do you go about matching them up? Or I mean, I guess you're looking for, maybe a metabolomic signature that matches with gene expression or something like that.
Yeah, terrific question. So we typically, in terms of the approaches, typically there are like 3 steps. The first one is to look at the technical validity of the analysis, especially when the sample size is small.
Mm-hmm.
For example, when we look at the whole genome sequencing, as you know, there's 3 billion bases in the genome. The chance that one make a mistake during the sequencing study, although that chance is small, the existence of a few mistakes would be very likely. Even in one a million bases, one can make a mistake. Then you still get like 3,000 bases that the sequencing would go wrong. So that's why we need larger studies and the comparisons between different studies to make sure that we're finding the same variant. So one of the preprint that just came out, this week, the reason that it only come out this week is just because we try to look at the multiple studies and try to see whether there is a consistency between them. So for that particular study, for example, that basically shows that at least in the severely ill patients, there seems to be what we call pathogenic or likely pathogenic variants that relate to neurological functions in the patients. So the hypothesis that one then can potentially generate would be, whether it is this type of neurological function related genes that are more relevant to ME/CFS, or it is more relevant to the severity of the disease.
Right.
So that those remain to be tested, but at least that sort of give us the direction to look for. And then the other side would be, whether we can actually do something about it, that can modulate those, genes that are involved in those pathways. So those are the sort of 2 directions that we're looking at. So this is about reproducibility. The second approach that we look at would be exactly as you said. You put the findings into the context of the collective knowledge of of biology. So because there's so many biological studies, and early on, I think this was late 1990s, a few graduate students in Ron Davis' lab and a couple of other labs at Stanford had this idea that if we collect all those knowledge together and put it into a database, so whenever we have a new study, we would compare what's found in this study with what people have already found in the past in terms of molecular mechanisms. So we call that knowledge-based network analysis. So we've been doing that for a couple of decades, and that seems to be very helpful. So if we have the metabolite data, we would then look at are there any differences in the genes that relate to that metabolite? If the expression of that gene is changed, then that might be the place that we should look further, because you have not only the gene information, evidence, but also the evidence from the metabolites. So that's sort of the way that we try to analyze this data. For the past couple of years, we start to do the third way. We call it network medicine, just because ME/CFS, comparing to other well-studied diseases, is much understudied. So the idea would be, if we just use our existing findings and compare that with the findings in all those well-studied diseases, can we actually answer the question, which disease would look similar to ME/CFS, just based on the incomplete findings that we currently have, and then look at the commonalities, among those genes that share the same, for example, symptoms as ME/CFS, and if there's a set of genes that underlining the mechanism of this well-studied diseases that's similar to ME/CFS, then perhaps we can make this assumption that we should study those genes, more carefully. And I guess another sort of a jump of leap of faith would be, if there are already drugs that are, proven to be effective in those diseases, perhaps we can think about testing those drugs in ME/CFS. So, all those work have been done for a couple of years, and we actually have some results that we're thinking, we're actually debating ourselves, whether we should publish those or do more work about it. A reason is, I actually need to learn from you. All the treatments would potentially have side effects. So, I'm actually not sure what's the best way to publish those, but those are the results that we currently are generating.
I guess that's always a worry, that somebody's going to jump on a potential finding that hasn't been
exactly
you're not sure about, and somebody's going to get hurt. Yeah, because people really want to get they'll do, as we know, just about anything to get well. It's still I think it's a brilliant approach to leverage all the data on other diseases in order to learn about ME/CFS. I really don't know what else you could do other than just plugging away with our small data sets. What about how much will the decode ME study help with that? Is that going to be a big help?
I think so. Especially from the genetics side, I think that would be really helpful. If anything, I can say, in terms of the genetic contribution, there's one school of thought that it is this relatively common variants. So those are the variants that not extremely rare, might be underlining ME/CFS. So that's sort of the underlying hypothesis of so-called genome-wide association studies. And as you know, Chris Ponting, who leads this Decode ME project, essentially work on this hypothesis. One of the ideas that we're testing that's a little bit different is whether it is also possible that different patients would have extremely rare variants Those are the pathogenic, likely pathogenic variants. The issue with those variants would be often they're very, very rare because there's a selection against it. And then for genome-wide association studies, there's a chance that those ones would be missed. So these days, people instead would do whole genome sequencing or exome sequencing. So that's the type of data that we're currently looking at.
So could you explain about whole genome sequencing and how it's different from like the kind of 23andMe or Ancestry.com analyses that we as patients do? Because that was a big thing in your latest paper.
So in we know that for human genome, there's 3 billion bases, right? So there are 3 billion places to look at the genome of a person. But then
3 billion possible mistakes.
Exactly. But then for genome-wide association studies, the particular tool that people uses these days would only look at a selected subset of hundreds of thousands to a million typically. So in other words, there's a 1 in 3,000 places that we actually look at.
Wow.
And then for the rest, we guess. So that's basically the idea of genome-wide association studies. So if the variant that really matters is a relatively common one, we typically can guess that well. If it is extremely rare, it's often very hard to guess unless you measure it directly. So that's basically the problem with using genome-wide association studies to look at those really pathogenic variants, just because you don't really test them directly. So the way to test them directly would be just sequence them directly. And these days, because as you know, the sequencing cost is substantially reduced. So I think the chance that we can look into those, we still have to look at those carefully, but the chance that we can look at them would increase substantially these days.
Yeah, I'm sorry if I'm like, I have 2 puppies here and they're chewing on different things.
No, I have a puppy too.
So if you hear like, yeah, that's what's happening. I'm trying to like stop them.
No, I totally understand.
Well, so is whole genome has that been done very much in ME/CFS? I noticed you did it in this latest paper, and so you identified 96 variants, possibly pathogenic variants that you thought might really be contributing to the disease. And six were found in more than 10% of the severely ill group. Was that surprising? I mean, did you expect more commonality, or is that actually a high degree of commonality, with regarding these kinds of studies? And then we'll go, because then you did something else which really kind of expanded the analysis greatly, but at least with these, which led me to believe that it really didn't matter that much whether they were shared or not. But anyway,
excellent question. So for classic genetics diseases, you would expect that similar variants or pathogenic variants that cause a disease. So when we saw the results, one of the things that we were thinking is, what's the chance you would have sort of multiple different variants that contribute to the same disease? That's part of the reason that we were trying to sort of link those genes together to see whether there are any commonalities there. And as you mentioned, our findings is that they seem to be, linked to each other and contribute to potentially the neurological, functions. And then the question would be, how likely a potential issue with the nervous system can actually either contribute to the disease itself or contribute to the severity of the disease. That's what we're testing right now. I'm not sure whether, let's say, patients with this particular type of variant would have higher chance to get ME/CFS, or only when the person has ME/CFS, this variants in the neurological system would actually contribute to the severity of the disease. I don't know the answer. That's what we're trying to figure out. And one of the ideas, as you already mentioned, is we're thinking of analyzing the decode ME data to see whether we can actually learn something about that. So that's sort of the next step that we're thinking about.
Yeah, I thought the paper was really interesting.
Oh, thank you.
You did a protein to protein interaction network, which I guess describes how the proteins that they bind together in the cell, and then the proteins are kind of the workhorse of the cell. So they do all these different things. So you looked at the network in the ME/CFS patients, and then you used a disease module detection algorithm to create a severely ill ME/CFS disease module, which had been seeded by these 96 genes, possibly pathogenic genes. And then you looked at and then you found that many of the genes have been associated with fatigue and cognitive problems. So that was good. That suggested you were on the right track there. And then you found that the module kind of intertwined with 6 neurodegenerative disease pathways. I mean, it really seems to be pointed to the neurological system. And when we're talking about the neurological system, are we talking about just the brain, or are we talking about the nerves, in the body as well?
Yeah, good question. Good question. I don't think we have actually that level of resolution at the moment, because as you mentioned, what we did was we basically this was actually based on previous work by different groups. People try to identify for each well-studied diseases, let's say Parkinson, what are the genes that might be causing that particular disease. So we can imagine that for each disease, there's a list of genes that are significantly related from either genetics or from genome-wide association studies we mentioned. And then you just compare this list, as you already mentioned, this list of genes that we found in severe ME/CFS with those diseases, in this case, a few hundred different diseases, and ask the question, what are the ones that show significant overlap? In other words, more than random. And it just turns out that the top ones.
Let it go. Sorry.
No, not at all.
They're starting to play with each other. Okay. Yeah, I don't know if there's one of them right there. Yeah, but anyway, I don't know if you can see that. So you're finding, it looks like clear links with
neurological
pathways in other nervous system diseases.
Yeah, and I don't know whether it's sort of a good news or bad news, because I thought that if the top ones are, let's say, more related to let's say, autoimmune diseases or inflammatory diseases, then perhaps there are more tools in the
Oh,
research world to study them. As we all know, the brain and central nervous system is harder, or even just the nervous system in general is harder to study because we can all take blood samples, but sampling the nervous system, especially the central nervous system, is very difficult to do. So, but at least that points to the possibility of the involvement of the nervous system. And then we will all have to work together to figure out ways to study it. If indeed, this particular finding is going to be, I still think it's a pilot study. It needs to be validated by larger data sets. That's why, I'm happy that you mentioned, for example, the Decode ME project. And we're currently also looking at the US OFAS and the UK Biobank
oh,
data sets to see whether we can reach the similar conclusion. But assume that some of this can be validated, then I guess we all need to think about how one might be able to study this. For example, as you mentioned Jared Younger before earlier, obviously imaging is one of the potential ways to look at the central nervous system.
well, as a longtime ME/CFS patient, I kind of like it when you find anything that's associated with other diseases. Maybe in the long term, I would say no. And we're talking about nervous system diseases, but you also looked at depression, and
Yeah,
you found very little overlap
yeah,
between depression and
yeah,
ME/CFS, the genes
yeah,
associated and pathways
yeah,
associated. Yeah,
yeah. Yeah,
yeah.
I mean, I know in other places what this is called, but in computational biology we call it sanity check. So
Uh,
you just if everything is similar to ME/CFS, then that would be something that's suspicious. So we would actually try to see whether there are things that are clearly not associated. So what are the diseases on the other side? And just turned out that on the other extreme side, as I mentioned, there are a few hundred diseases that we looked at. On the other extreme side, it is this depression. So that's sort of give us a little bit more confidence that our findings might not just be a technical artifact, because you
right.
could potentially imagine that our results show every disease is similar to ME/CFS. that must be something wrong with the algorithm itself. So the fact that the depression is actually at the other end of the spectrum, it actually give us some confidence that the results might be potentially meaningful.
Again, it just seems great that you guys are continually trying to prove yourself wrong,
Right, exactly.
as
That's
Ron
very
Davis would
important.
say.
Yeah,
Yeah,
exactly,
yeah.
exactly,
I guess a lot of people
exactly.
try and prove themselves right, and you try and prove yourself wrong. Uh, and once you've gone through those steps, you can feel more confident.
Yeah,
Uh, so so in the paper, you also did multi-gene set analysis. I guess using the several data sets, and that several interesting pathways popped up. So we have neurological stuff popping up in the first analysis, plus I guess some immune, and then boy, interesting that it popped up things associated with viruses and
yeah,
infections, Epstein-Barr virus, antigen processing and presentation,
yeah.
and the MAP signaling pathway.
Yeah,
Um,
yeah. So to be honest, we're still trying to sort of digest what this potentially means.
yes,
Um,
but yes,
so
but isn't this? I mean, it's popping up in long COVID.
yeah,
Epstein-Barr virus did. It's popped up in ME/CFS for so long. That must
exactly.
that must give you a little confidence that
Yeah,
and
I
interesting that
agree.
the same thing is popping up in these really very severely ill patients. These were people who were homebound. They spent 14 hours, I think, bedbound.
Yeah.
They were really sick. And yet these similar these
Yeah.
similar findings are popping up in them.
Yeah.
For me, I think that's encouraging.
Yeah. I agree.
You
I
know,
agree.
that
Well,
there's not something really strange happening.
yeah.
It doesn't seem like something really strange is happening that's different from the ME/CFS population. Or,
Right. Right. Right.
is that correct? These are
Yes.
just
Yes.
these are people. The disease process has just become more severe.
Yeah, yeah. Oh, I completely agree. And when we saw this result, I think we had sort of similar reaction as you mentioned. And then, we got to think about, what would be potential mechanism. I'm sure, you wrote a lot about the potential hypothesis. And, we're doing a couple of follow-up studies along these lines ourselves as well. One is in collaboration with Ron Davis, just to see, whether we can actually detect those viruses if they're indeed, let's say, reactivated. I
Mhm.
think Ron mentioned it, as you know, in at least a couple of occasions that we found in COVID patients, do see this reactivation, and in trauma patients, we saw that as well. So that is,
Oh,
that is one of
EBV
the things.
reactivation
Yeah,
in trauma
yeah,
patients? Oh,
yeah. So that is one of the directions that we're looking at, and then we're thinking, whether if indeed the reactivation is contributing to the disease, what could be potential ways that it does it? Is it really
right.
an inflammatory response, or it is autoimmunity? So right now we're actually testing both to see, which one is more likely. I'm sure you heard, for example, Rob Fire's idea of this autonomic shunt. So that would be, I guess, one lines of thought that if it is an inflammatory response due to reactivation, and the body's response to that would be to reduce energy consumption, because I'm sure until, let's say, 100 years, from today, calorie restriction, or maybe not 20, a lot of other places in the world, they still have a limited, food supply, to the population. So, for many, many, many years, if we are sick, during the recovery phase, we can't really get enough food intake. So the way that our body naturally, respond to that would be reduce metabolism,
Right.
right,
Right.
to keep ourselves alive. So I think, it makes sense if that's indeed the mechanism. Um, but we need to validate it, obviously. Another possibility would be, some of the groups already showed. I think one particular one would be Akiko's study. As you probably know, they had a preprint in June where they did this so-called passive transfer. So they transferred the immunoglobulin to mice, and the mice seem to have some of these symptoms, right? So that would suggest the autoimmune mechanism, at least in long COVID. And so then the question is, in MCFS, is this a similar process that's going on? So that's another angle that we're testing.
So yeah, so I know you're computational, but can you explain? I mean, it seems like it's really difficult to pin down these autoimmune antibody
Yeah,
problems.
yeah, absolutely.
Uh,
Exactly as I
what is
said,
going on there?
yeah,
Uh,
exactly. As you mentioned, I'm purely computational, but our interest in this is because uh, early on during COVID, what we and my collaborators did was we looked at patients around Boston who had complications after acute COVID versus those patients who did not have problems, after being infected by the virus. what we found was actually there's specific epitopes, there's specific regions of the virus that the antibody actually binds to. And this happens because, the evolution of getting antibodies developed is actually a individual process. In other words, that evolution in my body would be different than yours. And when we see the same virus, because a lot of this is just by random chance. So what we found was there are specific antibodies that target a specific region of the virus. In patients who had complications versus patients who did not have complications. So you basically test for that, and then we found out that those regions that seem to be significantly increased in patients who had a lot of complications actually share sequence similarities to our own body. Proteins in our own body. So that's basically made us to think that perhaps this, cross-reactivity. So the antibodies was developed against the virus, but it happened to target a particular part of the virus that actually share similar sequence to our own proteins. So therefore, the antibody that's generated would be able to bind to our own proteins, and that potentially cause this autoimmune problem. So that was sort of our initial interest in this. And, I was very happy to see, Akiko's preprint where they actually showed that when they extract immunoglobulins from long COVID patients and gave those to mice, the mice would develop symptoms. So that would suggest that there's something there in the antibodies of long COVID patients that actually can cause symptoms. So we're, then the question would be, which part of the, the virus, is it actually the SARS-CoV-2 virus, or it is, let's say, this reactivated virus? And if it is reactivated virus, EBV is one of them. You wrote a lot about this, HHV6 or, CMV, or, there are other ones that might be relevant as well. So because I'm a computational person, so we can just basically look at all the, known viruses, bacteria, and see what are the ones that might be candidates. And then, one can then go ahead and test those. So, if it is indeed a EBV, can we actually test those, antibodies? If it's indeed a bacteria, can we find those as candidates and test those?
You
Obviously, I can't do the test, but I give the hints.
You analyze the results.
Yeah, yes. Yeah, exactly.
Yes. But so it's like searching for an antibody that's reacting with EBV that would also be reacting with our human tissues.
Right.
And
exactly.
that's not as Ron would say, not a trivial matter, I guess, to find that antibody. But the search is on. Very good. Very good. Do you know if they have found that in multiple sclerosis? Do you have any idea? I know that's not your
Yeah. I mean, you probably know that paper that Mike Jensen at Ron Davis Lab published earlier this year, where they showed the similarity in terms of the myelin basic proteins MBP degradation in ME/CFS patients versus multiple sclerosis patients, and in their results, it seems like there is a similarity between these two. So they only look at the target in the nervous system, this MBP, which is and they showed that between ME/CFS patients and multiple sclerosis patients, the degradation pattern in this particular protein in the blood seem to be similar. So that would suggest that there's potential similarities if indeed there's a autoantibody that, as you mentioned, or as you indicated, that could potentially work on multiple sclerosis and basically digest that MBP protein as a result of this autoantibody in multiple sclerosis. Then there's potentially similar type of autoantibodies in ME/CFS that cause that same protein to be degraded. Whether it's exactly the same antibody, I don't know, but it seems like it might be working on the same protein. So there's certainly a number of studies that show these lines of thought, but again, I think we have to basically validate this rigorously, just make sure that there's not really a technical artifact, and make sure that in significant percentage of patients you can actually observe this particular observation.
Yeah, so many interesting possibilities. All we need is a lot of money.
Yeah,
really.
that's true. That's true.
Well, anecdotally, it's interesting that I've heard that some people have done well on Copaxone, which I think is a
yeah,
multiple
exactly.
sclerosis.
That would be one. Exactly.
Yeah.
And as you know, there's this idea of using IVIG immunoglobulin therapy. There's that study I'm sure you know from University of Berlin, where they did the immunoabsorption to remove. So there's certainly these lines of thinking. I'm sure you're very familiar with this because I read a lot of this actually from your blogs or your website.
Oh. Well, I hope I got it right.
Oh yeah. It's really insightful, and I think once a couple of weeks, someone in my lab who are also computational would come to me and recommend one of your publications on your website to me and say you have to read this. One example is we found that we suggested that 2 amino acid supplement that might help some of the patients, and they told me that your blog actually explained it better than they were able to, because they only know, or for that matter, me too, only know that particular little pieces of the whole picture, and we actually learned from your writing. So truly, I appreciate what you have done.
Oh, well, that's nice. I like to say that my understanding is like this deep, but it's like this wide. So I just cover a lot of stuff. So
yeah, I guess we all have sort of our own little area of expertise, and
yes,
that's just part of the reason that we need to talk to each other and learn from each other. And for that matter, I really learned a lot from your website in terms of understanding the disease.
oh good,
so truly I appreciate what you do.
No, thank you, that's great to hear. It's great to hear. Yeah, in your latest study, two other things really popped up. Well, you did find cortisol. You did find a cortisol pathway enriched, I guess it is.
Yeah.
And found evidence that cortisol plays a role.
Yeah.
You also found a possibility of estrogen.
Yeah.
Boy, I think that is with regard to female predominance. Can you talk about how that might affect ME/CFS, an estrogen issue?
Yeah. So the sex hormones together with cortisol, on one hand, really sort of make us to think more about, as we talked about earlier, the adrenal gland. Whether we should actually look more into that. Partially because the first author of that paper is you might know is actually a patient researcher. So I know him personally as well, and he has I guess a severe version of the adrenal gland problem. So that sort of made us interested in that particular problem for a while. And you probably have read also Akiko's preprint. I think it was published this year on the sex hormones in long COVID, where she showed that the sex hormones apparently has most difference between patients versus controls, along with cortisol. So, in her preprint, so that sort of made us think that, because as you know, a lot of these hormones plus cortisol are at least partially made in the adrenal gland. And if the HPA is, the upstream regulations seem to be normal, then a sort of place to look at would potentially be the adrenal gland. And then the question would be, can we actually formulate since I run a computational lab, as you know, so the question would be, can we actually formulate a computational problem to test this? So a way to do this would be, there are diseases that that are caused by this type of deficiency. Then we would use similar methods as we discussed earlier to see how much similarity is there between the symptoms in those diseases versus MCFS, as well as molecular studies in those diseases comparing to the studies that have been done in MCFS. I hope that one day we'll meet again and we can discuss the results. How similar these are, but it is something that we're thinking of looking at.
Yeah. Another really fascinating thing in the paper was finding this Adam TSL2 gene in 45% of the severely affected patients, and it's rarely found outside, in the general population, and it plays a big role in the degradation of the extracellular matrix, i.e., connective tissues, and has hypermobility. And from what I've heard, connective tissues also include the blood vessels. I mean, they're just all over the place. Of course, there's blood flow problems. Boy, that really popped out there.
So for that particular variant, it is an amino acid change. So as you know, some of the variants, they basically disrupt the protein. So if this is a protein and the variant is there, and then basically all the amino acids after that would be wrong. So for those type of variants, for sure that that protein wouldn't function. And those are typically the pathogenic ones, right? But for this particular one, I think it's a valent to isoleucine, if I remember, isoleucine variant. In other words, it's not very clear how how pathogenic that particular variant is. But as you mentioned, the interest is mostly that it seems to be quite abundant in this set of 20 patients. So the question would then be, if you take another set of patients, would we expect a significant percentage of them would also show this particular variant or not? So it needs to be validated. At the moment, we don't know how pathogenic that particular variant is, because we know it's just a change of one amino acid. The rest are still the same.
It
But
kind it
then on
kind
the
of
other hand,
gives you
yeah.
a starting place. It would
Exactly,
seems.
exactly, exactly. And as
Yeah.
you know, EDS certainly is of a great interest. So that's part of the reason we put that in the discussion session of the paper. Just people can start testing, whether that indeed play a role or not.
That would not be something you would pick up in a 23andMe test,
Yeah,
or
for
would
that,
it?
yeah, good question. I'm not sure.
Uh-huh.
Partially just because that particular variant is not extremely rare, so there's a chance I think a typical test would have picked it up
Okay.
for that particular variant.
I'm just gonna read it out here. RS, if anybody wants to look, RS3576780 That's the I guess that's the variant.
Yeah.
In the Adam TSL2 gene. So if you wanted to explore this more, how would you explore it? You just try and get a bigger sample set? Has this variant been looked for in other studies?
Excellent question. So you probably know the genetics of the EDS is not you know uh, very much well studied yet. So there are certain genes and gene variants that we know would be significantly associated with EDS. But then there are other genes and variants that we really don't know how much they contribute. So that's why I'm saying that for this particular variant, whether it would contribute to the disease or not. At the moment, I don't have the confidence one or the other.
Mhm.
But then, in terms of research, what needs to be done would be, if we test this variant in a larger cohort of MCFS patients, if we also see the same variant, then that potentially would indicate that that variant indeed is associated with the disease. At the moment, I don't know the answer, but it definitely need to be further studied.
Oh, it's very interesting. It's very interesting. So could you just give an update on the I'm really keeping you a long time, but on the muscle biopsy study,
Yeah,
how that is going?
yeah. So I have to say, David Systrom, who you probably know, is the person that leads that study, and his group really has done a fabulous job in getting that going. Uh, uh, Last time I heard, there were at least 10 patients that have went through this 2-day CPAT study with muscle biopsies. And if I get this correctly, the current plan is to have 50 patients. So hopefully the sample size is large enough to allow a robust analysis of the muscle. And I'm sure the reason that I think it's really significant, especially from metabolism perspective, is that most of the studies to date on metabolism is done on circular, blood samples.
Right, right.
So the metabolites that we look at, they're generated somewhere, and then they're consumed somewhere else. But we really don't know where it come from and where where it is going. But then on the other hand, we know muscle, especially during exercise or physical exertion, uses most of the energy. So definitely looking at muscle, especially with respect to post-exertion malaise, we all know would be very insightful. Um, as you know, we looked at muscle biopsy data from the NH study, but that study is essentially analyzing muscle biopsy at a baseline. So,
Right,
we
right.
really don't know, what would happen if there is an exertion. So it is really, I think, the work at the Dr. David Systrom's place that make this happen.
Yeah.
Ron Tompkins, before he passed away, I think was the original person that sort of put the proposal together to try to get this project started. But unfortunately, he passed away, as you know, in 2022. And I'm just happy that eventually we get this project started, and hopefully next time when we speak to each other, we'll have some preliminary results to show whether some of these findings that for example we found from the NH study can indeed be validated using this larger data set, and if so, that can potentially point to the specific dysfunctions in the metabolic network that we can then think about treating.
Ah, well, how good of Ron and how typical to leave us with such a potentially groundbreaking study. I think of, Maureen Hansen's lab, which has shown,
Yeah,
that the metabolites are not kicking in, the proteins are not kicking in. There's signs that the muscles aren't recovering things aren't recovering
yep,
properly. And now you're actually looking at the muscles
right,
themselves before and
exactly.
after exercise. God, it's a great study. It's a great study.
I completely agree with you. We're all excited, and hopefully we can find something very important from that study.
Yeah, yeah. If you're listening, sign up if you can. That'd be great. Um, um, so I wanted to ask about so you created this. The study was actually called a severe illness big data
Mhm,
study, I believe. So you created this huge
mhm,
data set, and now it's available to other
mhm.
researchers. Are you
Yeah,
are you
yeah,
seeing people get in there and
yeah.
explore?
Yeah, so we shared it with a number of groups, depending on, as you know, for example, for genetics, it's a little bit more sensitive, uh, yeah, comparing to, let's say, yeah, gene expression data. But we shared with, I think, pretty much all the groups that uh, reached out to us and are interested in exploring further from the groups in Canada, groups in Australia, to a number of groups in the U.S. And as you probably know, they each one of them look at the data a little bit differently and
Yeah.
generated their own hypothesis, which I'm happy to see. I would think that with the publication of the NH study, that provides really a complementary data set, because some of the things that we looked at in the severely ill patient study and the NH study haven't really looked at, for example, whole genome sequencing for one reason or the other. But then some of the studies that the NH group did, which are extremely useful, for example, the muscle biopsy, we didn't get approval to do. So, yeah, putting all these type of studies together, I think would definitely help us. You already mentioned Murray Hansen's lab. They have a large cohort, and they're looking at multiple, uh, type of analysis on that cohort, as you know, as well. So putting all these data sets together, I think would definitely help us to figure out, what would be the molecular mechanism of MCFS. It could be just one mechanism. It could be, diverse.
Sure, sure.
So we'll just have to see.
Yeah, 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 yeah. I just have a general question
Mhm,
about artificial intelligence.
mhm,
So artificial intelligence, it's become a really
mhm.
big thing. It finally hit the public. Um, and I use it. Actually, I use it.
Yes,
I just
we all use this
ask
phrase.
simple questions.
Yes.
Um, um, but I mean, you have you been doing artificial intelligence for a long time?
Yeah, for a
We
while.
had
Yeah,
we had a blog on somebody who um, he compared findings from other diseases. He used artificial
yeah,
intelligence. So I mean, is this something that's been going on in your lab for a long time, or is there a
yeah.
distinction between artificial intelligence and kind of things you're doing? I'm just curious.
Absolutely. It's a terrific question. I think in the future, everything would converge.
Mhm.
So the current, um, you know, uh, consumer-facing, large language models, for example, would eventually become sophisticated enough. I think in the next decade or two, that can, you know, to some extent help a researcher
Okay.
or just
Okay.
everybody um, to basically do research from the computational perspective. At the moment, exactly as you alluded to, I think to me it's more a you know uh, in the context of research, it's more a teacher or a how do you say this?
Way to
Either
explore things.
either college or college I think college probably is uh, either high school or college level of
Wow.
of teacher.
Uh-huh.
In other words, if I don't know that field, what I found out was more often than not the information. I mean, you probably have the same experience as well. More often than not, the findings that we get from large language models would be relevant, and often they're correct. But at the level that I just don't know the field, for
Okay.
example, what BDNF does. If we look at the response from large language models, the majority of the answers would be correct, right? It still has some issues. What is currently lacking, at least in our experience, is that if you want to dig deeper, you want to ask the question, what are the evidence that suggests that BDNF is involved in, in ME/CFS? That's the place that I think the answers get less often to be correct. So the deeper type of analysis, I think, in the current, published large language models, it's still lacking that reasoning. And often, the individual researchers would, have to basically go to read the publications. For example, the large language model sometimes would recommend and go from there. And you read the papers themselves, or you feed those papers back to the large language models and help it to get a better answer. So in my experience, that seems to be the the current state of the art. So in-house, what we do is exactly as I described. We use some of these large language models locally, partially because of this patient protection issues. So you can't really send, for example, patient information to ChatGPT, right? So it has to be protected within the firewall. But then you try to do this iterative analysis, getting the wrong answers in the beginning, but then you try to feed it with more and more, enriched, additional data to help it to come up with the right answer. But then during this process, obviously, the researcher themselves would also read the paper and would also come up with the answer, right? So in our experience, at the end, it seems like these two answers are very similar. So at
Mhm.
the moment, it takes more time for us to actually, feed the large language model to come up with the same answer, just because, as I described, then just you go to read the paper yourself.
Oh, gotcha,
So,
gotcha.
but that's just now, right? So
Yes, yes, yes.
this problem would certainly evolve. So one of the projects, I don't know, you probably have heard in medicine. I think a number of medical schools are trying to do is to essentially train specific models. So I would only train model using high quality clinical,
Mm,
publications.
yes.
And because it is a common problem. If a problem has been studied for, let's say, 50 years, the large language model would basically take all the information in those 50 years and treat them equally, right? So
Um,
then,
mm,
so which means
mm,
that the new information
mm,
would have less influence than the old ones. But
ah,
that's sort of an intrinsic problem. But then, a way to get around that would be you only take the selected,
high
information.
quality.
So we
Yeah.
actually did that for CFS with the help of a few patient researchers in Open Medicine Foundation. So they basically picked, I think, somewhere around 1,000 publications, and we actually put them into that pipeline, and we currently use that ourselves. And I think with new publications, hopefully eventually it can be uh, intelligent enough and robust enough for us to look at some of the uh, you know questions with respect to this disease.
Oh, interesting. So that analysis is not ready for publication, or are you guys
It's a good
planning to?
question. Good question.
Yeah.
I probably need to speak to the person. So there's postdoc that works on this, and I need to speak to him to see whether how comfortable. I think part of this is sort of our own limitation. So I think as a computational person, we're intrinsically more conservative because um, just because we can test so many things at the same time. As you know, computation is typically cheaper than doing experiments. So, we're sort of trained to um, always think about, accidental findings. So if you
Right.
test 1,000 things, it's bound, we're bound to find something, positive. And, as wet lab scientist, you often can't do 1,000 experiments at the same time. So it's not something that they think about all the time. But for computational person, we test 1,000 hypotheses at the same time, all the time. And then if you find something positive, if we just go to publish it, then we're just going to add too much noise to the literature. So that's sort of we're intrinsically trained not to do that. But then, a side effect of this is often we're potentially be too conservative. We have to wait until we have sort of enough confirmation um, to actually publish something. So it's sort of a trade-off that we have to, and I totally understand that we certainly own our patients the updates on what we have been doing, and what might be something that potentially would be exciting, but I think
Right.
it's a
It's a fine,
trade-off
it's
we
fine
just
line.
have
It's
to.
a fine line.
Yes,
Uh,
yes.
yeah, yeah. Well, it certainly sounds like, I mean, you have a lot going on, testing all sorts of different hypotheses, um, looking at different aspects of the big data set. And it sounds like you have a good number of patient researchers, people who are patients who
Yeah,
are assisting you
yeah,
in your research.
yeah, yeah.
Yeah,
Yeah,
yeah.
as I mentioned, as a computational person, I really feel, especially when we study a real disease, I really just know too little. And when we only look at things from the computation perspective, we often, uh, identify things that we think are important, but just because our lack of understanding of the real disease.
Right.
So I think one uh, contribution that I truly appreciate from patient researchers is their opinion actually keep us grounded. So we have like meetings every 2 weeks with patient researchers, and they give us advice based on their own experience and their perspectives, and I found that really helpful. And we're publishing some of our results together with some of the patients because they really contributed significantly. And not only that, we actually had the opportunity to work with some of the patient researchers, for example, Mata Aki, you might know, who developed this treatment survey that we found very useful. Just
Yes.
because on the computational side, you can sort of based on genes, proteins, predict what treatment might be useful, but we don't really know, whether there's any ground truth there. And the results from her survey certainly tell us the right direction to pursue. So I really admire her dedication and the ingenuity in making that happen. So there are a number of examples that I really appreciate that patient researchers help.
Right, very nice, very nice. Well, one last question: is there anything in particular that's exciting you, really exciting you in the field or your work, or
Yeah, I don't know what's probably to say because for two reasons. One is, as I mentioned, we sort of use that computational pipeline and identified a few candidates, and we're still trying to figure out how to test those because if it is a molecule that's new, how would one be able to discuss it publicly without really causing some of these unexpected problems. That's still something that we're thinking about. We're trying to figure it out, and I think we'll definitely have to go through all the testings. But we identified a couple of those. And as I mentioned early on, we're talking about those two amino acids, ornithine and aspartate, and I think those are sort of the I would think naive, harmless molecules. But then some of those molecules that especially potentially modulate the nervous system, there's always this consideration that maybe we have to test more carefully before we can actually make any, potential public announcement, that kind of things, or even get it published. So we're still struggling with that, but it is exciting from the computational perspective that we can actually identify something that might be worthwhile to be tested. The other part that's I think interesting for me, just from a computational perspective, is that since we looked at the muscle of ME/CFS patients and actually saw some of this difference between patients versus control, so that made us thinking, can we actually make the same type of analysis of liver and potentially brain? Because in terms of metabolic organs, those are the three sort of major organs in the body. Obviously, it's extremely hard to get samples directly from patients in the liver or the brain. As you probably know, for the brain, there's autopsies samples. But our thinking is we can borrow strands from some of these other diseases that we found to be similar to ME/CFS. For example, we talked about in the beginning some of the neurodegenerative diseases that indeed a sample from the brain are available. So we're thinking about doing that type of analysis just to be able to see, what neurological pathways in the brain, for example, indeed are potentially, perturbed. And then we can, make recommendations to people who do experiments. Can you actually design a label, imaging label to test that? So that's
Mm.
sort of the second, just as a computational person, that's what I found that, interesting. And I hope I would be able to update you at some point, of our findings.
Yes, yes. Well, it's great that you have some really strong candidates that you're looking at, and then I hope I hope you get to the point where we can hear about them,
Yeah,
and and if something leaks out every now, if it leaks out, then it leaks out,
yeah, you and I can always talk. Absolutely.
Yeah, yeah, yeah. Um, and boy, it's interesting that the liver, you're thinking about the liver, because
Yeah,
I know
exactly.
it's something I never really heard about much until the
Yeah,
last couple of years.
yeah, exactly,
Um,
exactly. And I
yeah.
have to say, one of the early person that mentioned this to me is Thomas, who's a you probably mentioned. I think you wrote about him. He's a patient researcher in Greece. Uh,
Oh, yeah.
you actually wrote about him, I think, in Health Rising.
Yes.
Yeah, so I had the opportunity to work with him for the past year, and he was actually one of the early ones that mentioned this to me. So we sort of started to think about how to do that computationally.
Oh,
So
really? Oh,
yeah,
great. I was hoping I was
yeah,
hoping somebody would
exactly.
pick that up. Yeah, yeah, yeah. Oh, very good. Very good. All right.
Great
Well,
meeting you
yeah, it's great
again.
seeing you again. And
Yep.
yeah, thank you so much for taking the time. I
Sure,
know
absolutely. It's such a
this
pleasure speaking with you.
oh, I likewise. Likewise. Yeah. I imagine it's well into your dinner time now. Uh so but anyway,
Thank
thank you
you.
so much, and continued good luck
Thank you.
with all the work. Yeah.
Thank you. Thank you for what you do as well. Thank you.
Oh, thank you. Yes. Well, you make it possible by doing all this research.
Thank you.
Then I can study it and write about it. Okay. All right. All right.
Thank you very
Thank you
much.
again. Okay.
Bye bye.
Bye-bye.